Okay, hi everyone. So yeah, I'm talking about a key component of my program, which is developing predictive diagnostics, and it's great to be able to discuss it with you today. So I'm based at the Palmer Research and Extension Center, which is pretty close to the Oregon border. It's a pretty diverse place to grow crops. Apparently there's 104 different commodities grown, but my program mainly focuses on vegetable diseases, although we are a diagnostic lab as well, providing a frontline service. If people don't know what their plant disease problem is across all crops and even home gardener stuff, we offer a diagnostic service. We have a field program where we do a range of field trials across several vegetable crops and also hops and sweet corn. And my research program does focus on new diagnostic methods, characterizing plant pathogens, and overall using that to predict disease. Hopefully we can get ahead of the disease curve. So this is what I mean by predictive diagnostics. So typically when a diagnostic, sorry, this advanced slide is quite a hair trigger at the moment. Typically when a sample comes into the diagnostic lab, we can identify the causal agent within a week or two, sometimes quicker if we have an assay for it. We can deploy the correct treatment to manage that. And it's pretty useful. It will reduce the impact of the disease, lower the disease curve, but it also helps us with our disease intelligence, what we can do better next year. But with predictive diagnostics, it's an example of precision agriculture or horticulture. It can aid in our planting decisions, when to deploy that spray program. And hopefully we are getting ahead of the disease curve and we can minimize that impact, sometimes completely, hopefully. And this is good. It can reduce your losses and also pesticide inputs. Now, it's not a new concept. Predictive diagnostics is a relatively new phrase, but in terms of screening soil or screening seed, this has been done for a long time. Nematode soil sampling is pretty well established. There's lots of seed testing labs doing those traditional conventional methods for seed testing. Visual inspection of potato seed tubers has been done for a while, and obviously they've been using antibody-based methods for virus indexing. And then there's insect trapping as well. However, the advent of real-time PCR, quantitative PCR, TACMAN, has created a whole wealth of new possibilities for predictive diagnostics. And it's this very sensitive method in terms of quantitative PCR. It's its sensitivity, it's its specificity, in that we can specifically test for pathogens which we couldn't test before it was around. And then it's also the versatility of it, not just the science, but the ability of it to be deployed in diagnostic situations has really created a whole load of new possibilities for predictive diagnostics. Now, when we're developing predictive diagnostics, it really is an interdisciplinary effort. So, I'm a plant pathologist who does molecular biology, and you really have to sort of get into all there's an overlap of perhaps three main disciplines here: taxonomy, epidemiology, and molecular biology. So obviously we need good tests, but the taxonomy of what we're testing for really needs to underpin that. And a lot of the diseases we are testing for, specifically with soil, are species complexes. So we need to understand those species complexes. And then we do need to know the epidemiology. So, how does environmental conditions impact the disease development? We can detect the pathogen, but we need to sort of give the risk in terms of what the weather will do. We also need to look at a source of the pathogen. So it's great having, say, a spore trapping network, but if the pathogen is also coming in on seed, we might need to know the relative importance of that source of inoculum as well. And then there's also sampling strategy, especially with seed and soil. We need to be aware of our sampling strategies. So I'm going to talk about the 3 predictive diagnostic projects that we've been working on in the Palmer lab over the last 5 years, and that's spore trapping, soil testing, and seed tuber testing for potatoes. So why do spore trapping? Well, we can detect spores of a pathogen prior to symptoms, so we can get our sprays on in time. Sometimes, the pathogen is not in the area, we don't need to apply. Sometimes we need to know the pathogen is there earlier than we think, because it's best if we deploy any fungicide prior to actual symptoms. We need to apply it when spores are in the area. It's also useful for monitoring seasonal trends of the pathogen, so that would help with developing a spray program. Also distributions of pathogens, perhaps ones with a more restricted distribution, that would be useful information. But overall, it's for early warning, early deployment of treatments, and reduce losses and increase profitability for the grower. So what drove me to get involved in spore trapping in Idaho was Phytophthora infestans. This is the #1 potato disease worldwide, but in Idaho, because of the great growing conditions we have, it's considered a transient pathogen. Some years we have it and some years we don't. And growers don't want to be wasting a lot of fungicide spraying for it when it's not a blight year. Like in the UK where I'm from, a weather-based forecast is pretty useful to predict when you want to put a spray on. However, in Idaho, that's not always the case because we have such low disease pressure. Weather-based forecasts alone are not that useful. So the Idaho Potato Commission and the industry about 5 years ago were a big driver in setting up the spore trap network, initially for potatoes, but we've been able to expand into other crops as well. So what are the options for spore trapping for plant pathogens? Well, perhaps one of the most established systems or platforms for spore trapping is the rotor rod, sometimes called the spore stick. This is the one on the left-hand side of your screen. This is essentially 2 rotating arms which have got some lubricant on which trap the spores, and the horizontal bar spins very fast. It creates a vortex and spores get stuck to those vertical poles on that horizontal arm. And then you take those rods and then you extract DNA from them and you can test them for whatever pathogens are present. There's also a burr card with the tape drum in the bottom. It has a suction device in the top, and spores get stuck to a rotating tape drum, which has been around for a while as well. However, messing around with lots of tape, even though it's a low-cost, low-tech method, and you can actually see the spores under a microscope on the tape, it can be quite difficult, especially if you want to do a high-throughput result and get the results out quickly to growers. The other issue with the rotor rod is sometimes those rotating arms and the lubricant can get a lot of pollen, a lot of dirt on the sampling pole, and that can impact your results as well. And also you have to kind of change it out every day or every 2 or 3 days if you want to get good results. You can't just leave it for a week and come back to it, because they will just get clogged up. There's some more expensive or completely automated traps that have come out more recently, both offered by British companies, one offered by the Burkhardt and the other one operated by Optogene, who make the lamp devices. Both of these sort of are essentially some kind of pipetting mechanism within those boxes, and they do a lamp within the box and then send the results back to you by phone or by email or something. They're quite expensive, and you're kind of locked into their chemistry, so they're not adaptable as say other systems. But if that is the cutting edge, and to have something that can just be left in a field and send a result back to you would be very beneficial. More recently, a company called Scanit Technologies have released a spore cam, and they're renting these out. And I think this technology is very promising. How this works is the spores are sucked into the device, and they end up on some kind of cassette where a it is photographed, and using image recognition, they will recognize the spores and give the alerts that way. That's obviously pretty handy because you don't need to have a real-time PCR assay to identify it. However, this technology is pretty expensive at the moment and it's still early days. So what we use in Idaho is we use a Burkhardt automatic multivial sampler, and we're also using Spornados. So the Spornado is a passive spore trap, and essentially it's a funnel on with a wind vane, and it points into the wind, and then at the back of the funnel is a mesh, and it traps the spores on that. And then you take the mesh out once a week, extract the DNA, and you get your results. The Burkhardt automatic multivial sampler is a little bit more sophisticated, and it's 10 times the cost as well. But it does have the benefit of there's 8 vials on a carousel. The spores are actively sucked in using a pump, and the spores end up in one of those 8 vials. And then it's battery powered and solar backed up with a solar panel. And you can visit them once a week and get 7 individual days worth of data. So it's a pretty useful platform. And that's the one our main one we use in Idaho. So as I explained, even though the project was initially about late blight forecasting, because late blight is so transient in Idaho, it wasn't a very good model to actually develop a system on and evaluate that system. So we actually used a sugar beet disease in our first instance to get a feel for spore trapping, and we used powdery mildew. This is consistent every year at Palm, and we see it and we know we're familiar with it. And we do have a sensitive TACMAN assay to do the real-time PCR for that. So Carlos Pizzolotto, who was my postdoc who did this, he set up several traps on the side of a sugar beet field to evaluate which one kind of worked best, or hopefully confirm that the Burkhardt was the right choice. And the 5 traps we evaluated, obviously there's a Spornado and the Burkhardt, but on the left-hand side, we evaluated a very simple trap, basically an encapsulated glass slide with some lubricant on and a pump. So what happened is the pump sucks in air through that glass slide, or past that glass slide, and the spores get trapped on that. So we evaluated that. That we could only do for 15 minutes a day, but that was one of the ones we evaluated. We also evaluated the rotor rod, because that seemed to be the industry standard. And then we made our own version of the Spornado, which we christened the Cheapskato, but we made it out of a funnel we found in the lab. And rather than using a dry membrane to capture the spores on, we actually made our own. We actually had a plastic slide at the back which had some lube on. So it was comparing the sort of a dry membrane with a passive spore sampler with the Spornado, where with more of a moist one with the Cheapskato. And these were the results we got. So it's not a great slide, but we evaluated it over several days in late June to early July, well, mid-July, when we knew the spores were coming, sort of developing in the area. And it was quite promising in that the cyclone multivial, detected it on the first day, as did our coated slide trap as well. And what was quite encouraging was even though the coated slide and the pump was only run for 15 minutes, we did get some detection on that first day, although at low levels. But for 15 minutes, it was quite a good result compared to the others, which were running for 24/7. The spore stick gave us similar results to the Burkhardt multivial. It was disappointing we didn't get great detection with the Spornado back then. So it was about 2 weeks after our first detection with the Spornado. It was 2 weeks after the others. But if you notice, which is the gray line, the gray bar, when we got to sort of higher inoculum levels, the Spornado actually outperformed some of the other platforms. So although the Spornado is advantageous in that it's very cost effective, but it might be better deployed where you've got higher disease pressure for some of these pathogens compared to Idaho conditions. So we were pretty happy with the Burkard multivial, and that's like our platform of choice now. And you can see with sugar beet powdery mildew, we get some good predictions of disease. So we always seem to detect our first spore 2 to 3 weeks, maybe even a month before we see visual symptoms in the field, and several weeks before, well, a week to several weeks before we see visual symptoms in the wider area. So we, with industry support, we set up a Burkard network. We've been sort of deploying since 2018, something like 10 to 15 traps throughout Eastern Oregon and Southern Idaho, all the way from Palmer, all the way up to past Idaho Falls and Rexburg. And once a week, in the months of late June to early September, my crew go and collect vials from Western Idaho. And then we have some industry partners who help us collect them in the Magic Valley as well. And then I have colleagues at U of I who help collect them in the East Idaho. And all these traps, spore trap samples come back to the lab on Monday, or the industry ones, if they're shipped in, come in on Tuesday. And then in the lab, we use Kingfisher MLs to help us with DNA extraction to speed things up a bit. And typically within 24 hours, we can get the results out. And we send the results out to a weekly email bulletin. Sometimes we've used a website, and we're actually working on a better website to have a sort of a data archive for these spore trap samples going forward. So, like I said, we didn't, we don't see late blight a lot in Idaho. So it wasn't until 2019 where the system was really tested in anger, where we did get a late blight outbreak. It was very contained, thankfully, just 8 fields in south central Idaho. We did get an early warning for it with a detection the week before at about a site 10 miles away in Sequoia. And what was really neat was we were able to sort of define the limits of that outbreak using the spore trapping. So what we actually did, as soon as we knew there was an outbreak, we actually got a spore trap and put it straight in the epicenter, as it were. And then we turned that on, and we got excellent detection with that spore trap, as you would expect. But then we were able to use the spore traps adjacent to that to sort of set up a risk zone where spores were present. So that's the blue circle, because the sample in Oakley in the south there didn't detect anything. So we were able to sort of put out, a better risk than we would have done without that spore trap data of sort of a definite area where there was outbreak fields, and then an area at risk because of spores. So a lot of growers were able to use that and not get too concerned if they were some distance away from that outbreak area. What was really quite neat with some of the spore trap data we've been finding is a good relationship between relative humidity and spore levels. I know that's been proven for late blight. Humidity does really govern the amount of spores there, but it was really nice to see it in our spore traps, especially when we get into grips with a new system. So the bars here are spore levels, and the lines are relative humidity. So we get that peak of humidity, and then we did get a flush of spores shortly afterwards. So that was quite encouraging to sort of validate the system, as it were. And then from that outbreak in 2019, and also looking at some of that humidity and spore data, we were able to develop some of our own models to improve the prediction of our system. So we think we did develop quite a complicated model, but the one we ended up using, we christened the CHIP, which stands for critical humidity period. And this is 10 hours at 80%. a relative humidity or more in July to September, we get a good match with historical outbreak data, and especially if you include spore trap data as well. This is built upon previous sort of simple model efforts. The first one is in the Smith period, which has been in the UK since the 1950s, and is pretty well used. And that's 90% relative humidity for 11 hours. And then that's been improved by the Hutton criteria in the last decade or so, where they took that 11 hours down to 6 hours. So in Idaho, I think we have we require less humidity, but maybe for longer to drive an outbreak. But this is just a good guide to improve our risk prediction for growers. So we have expanded into other diseases, and one disease we're working on is Stemphylium leaf blight in onion. It causes these kind of oval lesions, which can get colonized by Alternaria as well. And the lesions can sort of run down an onion leaf quite quickly. And in severe infections, it's been described that the onions are dying with their boots on. Now, thankfully, in Idaho and the Treasure Valley, we have good weather conditions. We don't have that much moisture, but in some wetter years, we've seen a lot of Stemphylium coming in. It's kind of in New York, and it's been quite a devastating pathogen where the humidity levels have been much higher. And of course, some of these isolates are resistant to some of the commonly used fungicides as well. And Stemphylium seems to get resistance to fungicides quite easily compared to, say, some other pathogens like Rhizoctonia. So we're working on that, and we have been able to use the spore trap samples. We set up one sample in an onion field, and we have been able to use the spore trap samples we've collected and to sort of understand the disease more. So in 2020, you can see we've got much more consistent detection. We did have a bit of a gap in the summer where we didn't see that many spores, say between the late July and early August. But then when the weather conditions got a little bit more humid, we did see this flush of spores come through. So it's pretty cool to see Stemphylium match up with the weather conditions there. In 2021, we obviously had the heat zone in the Pacific Northwest, and you can see in August, which is kind of like a critical period for the spores to infect and really take those plants down quickly. We obviously got a lot lower disease spore levels detected, and that kind of matched up with what we saw in the crops as well. Not much disease. So from this, we think we're going to set up several years of data, but we think we can use this to sort of direct which kind of spray program people should be thinking about. Obviously we don't want them to be spraying for Stemphylium routinely because there's a big problem with fungicide resistance, but they do need to spray routinely for botrytis. Which doesn't have much, it has less of a concern with fungicide resistance. So maybe we can use this spore trap data going forward to sort of highlight when maybe growers need to deploy an additional fungicide or change their program up a bit to consider Stemphylium. So just to summarize where we're at with spore trapping, I think with late blight, that really is the poster child for spore trapping. It's not just used in Idaho, that there's like several other countries where, like in Canada, where spore trapping is used routinely for disease prediction. And it's pretty good. An outbreak of late blight can occur any time in the season. So you don't want to be spraying all the time. So the spore trap data can direct when you want to sort of change up your spray program for late blight. I think for powdery mildew in sugar beets, the spore trapping approach is very useful to work out when the spores first appear for powdery mildew, and then you can plan the timing of your spray program from that first appearance. So some years it can be much later and you can save a spray. Some years it might need to be earlier. So the spore trapping is pretty useful for that. Excuse me. And then for Stemphylium, obviously people for onions need to have a spray program to manage botrytis, which is always there, but the spore trapping can inform you if you need to change it up to include Stemphylium as well. Now, there's two foliar diseases that are pretty important in the area, Cercospora in sugar beets and Alternaria solani. What we've noticed is we've got some really poor predictive power with that. So I think at Palmer for Alternaria, about a week before we see disease symptoms, we will get a detection in the spore traps. And then in other places, they've said they've seen early blight before we've detected the spores. Now, these pathogens, these fungi have quite large spores. And I think it's a big ask for the spores to actually make it to that spore trap. If there's like a localized part on the other side of the field, it's a big ask to get like a large spore size to travel and get into that spore trap. So there's a poor predictive power with some of those larger spored fungi. And in fact, for Alternaria solani, we've been using soil testing. So pre-planting, we actually test the soil for the presence of Alternaria solani. And we're doing it to decide where we're going to place our early blight trials that year, if that field's going to be a good disease pressure for the for early blight trials. But like I'm saying, we need to maybe for those larger spored fungi that there might need to be another approach for how we predict those. So that brings me on to soil testing and as a predictive diagnostic for plant diseases. And we've developed a method to recover DNA and RNA from a 300-gram soil sample. And that coupled with real-time PCR allows plenty of opportunities for testing. Now, this has been going on for like 15 years now in various other labs. So in the UK, FERA, and a couple of institutions in Scotland have been working on soil testing for potato diseases. And in Australia, they have been doing it for barley, called Predictive B, and then there's the Predictive PT system as well for potatoes. How I got involved in soil testing, and I did this when I was working in the UK, we actually developed it to test for white rot of allium. So it's the #1 onion disease worldwide. And just one of these sclerotia, which are like a quarter of the size of the tip of a ballpoint pen, can initiate disease. And once disease is initiated, it rapidly turns that bulb into more sclerotia. And then if you water, it can spread those sclerotia throughout the field. So you can actually sieve out these sclerotia, but with different soil types and also the time it takes to sieve out sclerotia, it takes quite a long time. So we developed a real-time PCR assay for it, coupled with a soil test, which could actually go up to 1 kilogram, but routinely we use 300 grams of soil. So we developed it for white rot. Now, at the James Hutton Institute, they've been working on it for black dot risk, and this is caused by Colotrochicum cacoides in potatoes. And this is really the poster child for any soil testing. So they were able to work out the amount of soil inoculum in their samples and develop some kind of risk prediction system. So between 100 and 1,000, you're starting to see a higher risk. You need to maybe deploy something. And then above 1,000, you're really in high risk territory there. So in Australia, the Predictive PT system, they've been offering this for a while. And this is an example of what their report looks like. So obviously with black dot, I've just shown you that the science there, you're able to, give a disease risk. But they've also been able to do that for Spongospora and Lordigini halax as well. And then under evaluation, which they're still developing their risk criteria for, various other pathogens there, and you see they separate it out. And we've been working on something similar at Palmer, we actually give the risk at 5 different levels. So not detected, trace detection, which is just 1 to 5 copy numbers, and then low, moderate, and high. And what you'll also notice is we're able to do this for both DNA and RNA. Because we're getting a lot of PMTV and TRV samples in, we adapted our method to be able to also test for those pathogens as well. So we are able to extract from one sample and do the RNA, the virus, fungal, and bacteria test as well. So this is how we give our results. We're probably going to improve this over the next year as well, but we also give an interpretation. So with Rhizoctonia, we're able to say it's not a significant source, but we should also consider seed. And we're able to, moderate trace, we're able to give some idea of what the risk is. But it's not always straightforward dealing with soil-borne diseases. So they do exist as species complexes. Now, Rhizoctonia is the key, well, the example I'm going to use here in terms of its species complex. So if you test the soil for Rhizoctonia, and just say it's Rhizoctonia, it's a bit false. You really need to be giving the anastomosis group. And the reason why, it has lots of anastomosis groups, but why do we care about them? Well, disease severity can vary widely between them. So like say AG8 will only affect cereal roots and the potato roots. It won't cause black scurf on potatoes or stem disease on potatoes. These AGs also have different growth temperatures. There's different fungicide sensitivity for certain fungicides. They might have a different inoculum source. So when we're developing these predictive diagnostic tests, we need to understand the species complex. So we did do a survey of what anastomosis groups are present in Idaho potatoes. Now, AG3PT, thankfully, was the predominant one. So it was causing all aspects of the disease, black scurf, stolon canker, stem canker, what have you. But we also recovered AG2-1 and AG4-HG2, as well as the odd occurrence of some other AGs. So AG, if you look on the next slide, if we look at stem canker, you can see that, AG3 causes a lot of disease, AG4 slightly less so on average. And then with AG2-1, even different subsets of it, even different strains within that subgroup can cause very different levels of disease. So we need to understand this if we're going to be offering a predictive diagnostic service. Are there other strains that can be causing disease? And if they are, what is the severity of disease there? So obviously, if say we did just test for AG2-1 and just presumed it was the same isolates were causing the same levels of disease all the time, we're going to have some instances where you're not going to see disease because there's 2 strains of that pathogen kicking around in Idaho, and they can have vastly different levels of disease development. We also need to understand our field sampling strategy for these soil-borne plant pathogens, which is what we're working on at the moment for powdery scab and PMTV. But we did some work with Rhizoctonia a few years ago, and typically when people sample soils, they might do what's called the lazy W and just walk in a W across the field. Now, I always recommend a grid simply because of this figure here. If we did the lazy W, the blue parts are much higher in Rhizoctonia levels. And you could see we've probably only got one sampling point on that blue level, which would really underestimate the inoculum in that field. If we did do a grid, we'd have probably about 8 of those high levels included. So I always recommend people do a grid sampling strategy for soils. And then we also need to understand other sources of inoculum as well. So Rhizoctonia seems to be pretty prevalent on seed. And these are actually some seed tubers that have come into the lab with exceptionally high levels of of inoculum on them. So if we also need to be offering seed testing and also understand what is the risk that pathogen is going to be coming in that on seed as opposed to soil, so we can improve that risk when we give the results to the grower. So we've been looking at seed tuber testing as well. And again, we've took this approach of developing a total nucleic acid method because we want to be able to test for viruses as well as fungi and bacteria in one go, so we can offer a cost-effective service. So we got a project to look at bacterial ring rot, and then as part of this project, we made sure the method we were developing could also get PVY, PMTV, and TRV as well. It's a cost-effective method, and we are validating at the moment with 146 samples we received between 2018 and 2020. And we actually use a 200 tuber sample, which we divide into 4 subsamples. And we take a core from both ends of the potato tuber. And it's really important to use the right corer. If you use a cheap corer you get from a supermarket, say, they pretty much fall apart, but you need to buy some expensive stuff from Amazon, maybe, or from a professional cook store, otherwise they just corrode and fall apart. So these are the preliminary results we got for Idaho seed samples. So we're still testing these at the moment. It's quite a long process to get through them all, to test for multiple pathogens. But we didn't detect any bacterial ring rot, Clavibacter, in any of the samples, but powdery scab caused by Spongospora was widely prevalent, as was PMTV and PVY. And then in our 2020 samples, we actually detected Dicaya, at least our lab, for the first time in Idaho seed. And then in terms of visual assessments, so we've assessed every tuber visually as well, and we're going to match that data back up with the DNA level. We did detect quite a lot of black scurf in those samples, in the majority of those samples as well. To work out how sensitive the method was, we used PVY and we blended infected cores with non-infected cores, and then we quantify that using a synthetic sample. And we were easily able to detect 1 infected core in 100, and our theoretical limit of detection is 1/100th of 1 infected core in 100. So we know the method is pretty sensitive. And this is the kind of results we can give out to growers. So if you look at the virus testing, we do give out an average, and then we equate that to what the equivalent is in terms of how many cores are infected, because we're comparing that with the established ELISA method. And one thing I would just like to draw your attention to, because we do 4 subsamples, you can actually get quite a range in that level. So if you look at, say, the sample 17 there, or even sample 14, it can be an 8% difference in that level of, in one subsample, in 4 subsamples from the same batch, they can vary by as much as 8%. So it's quite worrying when people are, say, taking an ELISA test and trying to make decisions based on 0.5% difference, where I think there's quite a lot of variability within any lot in terms of the amount of PVY that is present. So to conclude, I think it's a fun field to be in. It's a truly interdisciplinary approach to develop these predictive diagnostic methods, but it's not just about the test. It's great if you can have a really great specific PCR test, but you have to bring in other things as well. Your sampling. How can you make that test cost effective for the grower? Because they're not going to be paying thousands of dollars on a test. We need to get it in around $100 really to be cost effective. You have to really take a holistic approach in how you develop these tests, multidisciplinary, but also in how you interpret them as well. Like I said, with Rhizoctonia, yes, we can detect it in the soil, but that its prevalence in seed samples, we need to also consider that as well. I think with the spore trapping, it's a great example of proactive surveillance, and I would like to see more labs doing that, particularly diagnostic labs, because I think it's a great way of engaging with the industry. When we give out our email alerts, we obviously give them spore information. That's what people are tuning in for, but we're also telling them about other things we've come into the lab and other information. So it's a great way of engaging with the growers over the course of a season. And interpretation of those results is absolutely key, and to translate them into sort of management actions as well and advice. And that's what we're going to be working on over the next few months. So it takes a whole team to do this work. So I'd just like to thank my team at Palmer, and then also my colleagues, Kasia, Brenda, Mike, Phil, and Juliet, for helping with some of these projects. And then some external collaborators, Jeff Miller, Jonathan Whitworth, and Kawamu at WSU. And then industry collaborators and our various organizations who've supported this work in the past. So thank you for your attention, and I'm happy to take any questions.